Ladies and gentlemen, thank you very much for this invitation. Thank you, dear Pepe, for this introduction. The reason why the title of this presentation is Myth is simply because in my own experience, both in the ward and in the lab, I had such an enormous experience in the field, and I am sometimes surprised by naive conclusions proposed by researchers and clinicians in the field of Parkinsonism in general. So this talk will be somewhat provocative, right? So with me, would you accept to read what is written here? I will read it for you. Parkinson's disease is a movement disorder characterized by the classical triad, echinacea, rigidity, and tremor. The disorder is easy to identify, but the prognosis is always difficult to establish. The management of patients is usually well done, in particular with the use of motor scales. Symptoms start to be observed when the loss of dopaminergic neurons within the nigrostriatal pathway exceeds 50%. Symptoms are improved by levodopa, which could be toxic for neurons. Dopamine agonists are used in most patients to avoid motor side effects. High-frequency stimulation of the subthalamic nucleus can be helpful in late stages of the disease. The presence of Lewy bodies in the substance nigra of patient is characteristic of Parkinson's disease. The cause of the disorder is not known yet. The disease is rarely hereditary. The contribution of an environmental factor is discussed. We should soon slow down the progression of the disease as the main mechanisms of nerve cell death have been identified. One is waiting for the success of cell and gene therapies to cure the disease. So, ladies and gentlemen, what do you think about what is written here? For instance, what do you think?
This is okay. It's a fantastic summary of the disease.
Yeah. Except that nothing is correct. Nothing. I agree with you. This is what is written in our book. This is what we write for the students. Nothing is correct. So, because of time, in less than half an hour, I will choose some of these items and discuss them with you. First, Parkinson's disease, is a movement disorder characterized by classical triad. It's not Parkinson's disease. It should be Parkinson's diseases. Then I will show you that it's not a motor disorder. It's a neuropsychiatric disorder. And then I will briefly discuss the fact that the diagnosis is extremely difficult to perform, whereas the prognosis is easy to perform. Very briefly. So you see, Parkinson's diseases is in fact several disorders, as all of you know. We should say perhaps something like Parkinson syndrome with secondary cause, such as drug-induced Parkinsonism by neuroleptics. But in the so-called primary Parkinsonism, then you have two types of Parkinson's diseases. The most common are sporadic, 85% approximately, and hereditary in 15% of the cases. And in addition, you have these so-called Parkinson plus diseases, including complex disorders such as progressive supranuclear palsy and many others, and virtually very rare metabolic disorders such as gangliosidosis, alterations in the dopamine metabolism, for instance. So it's a very complex nosology in contrast to what we believed 20 years ago or 30 years ago when we were talking about Parkinson's disease, we should say, Parkinson's diseases. 2, Parkinson's disease in the classical form of the disorder is indeed a neuropsychiatric disorder, if I may say so. Why? Because in addition to the sensory motor symptoms that you know perfectly well, including actual symptoms that I will briefly discuss, you have cognitive disturbances of the subcortical or cortical type. You have eventually very severe emotional disorder. Depression and anxiety, I think, is present in all patients, with sometimes psychosis, hallucinations, and many other symptoms, such as the one described here. And as you can see, very few of these symptoms do respond to the replacement, levodopa replacement therapy. Why? Indeed, you see some are poorly responding, but some are not at all. For instance, cortical dementia. So it's, as I say, no, Parkinson's diseases are neuropsychiatric disorders. And it's so complex that for our scientific colleagues, I would like to show that each of the symptoms of this disorder is very, very complex. So we are, for instance, we are talking about bradykinesia. But bradykinesia is a very complex disorder, symptom. It includes slowness of movement. I'm slow. But it's also akinesia. That is the difficulty to initiate a movement. When the patient is sitting in a chair, he doesn't move. He's like that. And eventually his movements are relatively rapid. But the initiation is poor. This is reflected by the increased reaction time. Hypometria. When you ask the patient to do this, then after a few seconds, you see hypometria. It's not like that, it becomes like this, right? And this is true for all movements. This is why my famous predecessor, Jean-Martin Charcot, said that the Parkinson patient was condemned to prisons forever because he had to, by will, to compensate for the absence of automatic movements. Then disruption of sequential and concurrent movement. The patients can do this, as shown in London. You remember, Pepe? You can do this, but when you do the two movements together, then, for instance, reaction time is increased. And you see, this is perhaps related to movement, but it is also related to something. Why are these patients so slow? It's also, these symptoms is not only motor, of motor origin, it is probably of psychological origin. No, all the more since depression and apathy are seen in these patients. Have you seen a depressed patient who could move rapidly? No. A depressed patient is moving very slowly. So you see, simply one of these symptoms is very, very complex. And the reason why I'm saying all this is simply to say that a patient with Parkinson's disease is not like a rat with a 6-rotate dopamine lesion, which is itself a very complex model for Parkinsonism. Okay, so we are dealing with neuropsychiatric disorders, and I said that the prognosis was difficult to perform. This is not exactly true. You have disorders of that kind This is the motor disability with time. Some patients become very severe with time, others very slow. And the reason why these patients are severe under levodopa treatment is simply because these symptoms do not respond to levodopa replacement therapy. And the 9 symptoms that all clinicians need to briefly, it takes 20 seconds, briefly look at in these patients are what I call actual signs: frontal lobe-like symptomatology, eye movement disturbances, neck rigidity, speech disturbances, swallowing disturbances, abnormal posture, sphincter disturbances, and mostly postural instability and gait disorder. These are the actual signs which respond very poorly and eventually not at all to the medical treatment. And altogether, let's say that 15% of our patients do respond beautifully to the treatment. 15% do not respond to the treatment. And most of the patients start to respond quite nicely, but the response to the treatments becomes poorer and poorer. So for those who are interested in pathophysiology, this is a simple schema, I think, which summarizes the whole thing. What is the characteristic of Parkinsonism? The characteristic is the degeneration of the nigrostriatal pathway originating in the brainstem and projecting to the basal ganglia, mainly the striatum, right? Everybody knows that. So this is the lesion of the dopaminergic nigrostriata, as said a long time ago by Ole Ornikevich, is the main feature of Parkinson's. It's part of the definition. If a patient has solely this type of lesion, he's responding nicely to levodopa treatment, of course, which is a replacement therapy. But most of our patients have lesions that you can call in parallel. That is, patients are treated with levodopa. Echinacea, rigidity, and tremor do respond quite well to treatment. But unfortunately, because of other lesions in the cerebral cortex, in the brainstem, in the basal ganglia, in the cerebellum, etc., then these symptoms are caused by lesions which are not of dopamine origin. Therefore, they do not respond to levodopa treatment. And this is explaining all patients with the so-called Parkinson plus syndromes, in particular, with all these 9 actual signs that I briefly described to you. This represents 70% of the patients. At start, these are the predominant lesions, but with time, additional actual signs do appear due to non-dopaminergic lesions. And in the last 15% of the patients, lesions that you may call in series are seen in patients. And in this case, these lesions, non-dopaminergic lesions, are located on the output of the nigrostriatal system, downstream the striatum. And in this case, if you can give as much levodopa as you can, the dopaminergic transmission at this level does not occur because there are lesions in series downstream the striatum. This is what you see with PSP patients, for instance, progressive supranuclear palsy patients. If you keep this schema in mind, I think that's enough to understand Parkinsonism in general. Okay, so we've seen that it's a complex disorder. The nosography is very complex. It's a neuropsychiatric disorder, not simply a movement disorder, and we try to explain why. I will briefly tell you about, because of time, the other. The management of patient is usually well done in 50% of the cases. Usually it's a catastrophe for me, at least in the 50 other percent. With the use of motor scales, I think that in clinical practice, motor scales are totally useless, except for research purposes, of course. Symptoms start to be observed when the loss of the exceeds 50%. That's a nonsense. That's totally stupid. But I have not the time. In the discussion, if you are interested, I would be delighted to discuss this. Symptoms are improved by levodopa, which could be toxic for a neuron. Levodopa is not toxic. This has been proven several times in vivo and in vitro. And with Rita Reisman in our lab, we were even able to show that it's a trophic factor. High-frequency stimulation of the cerebellum can be helpful in late stage of the disease. As you have perhaps seen, there has been a French-German study showing that patients could be operated very much earlier during the course of the disease with caution, because this is a research paper showing that indeed after 7 years instead of 14 years, patients can be operated, but you need to deal with a lot of expertise before doing this. Anyway, so now I will come back to this, the presence of Lewy bodies in subnuclear structure of Parkinson. The cause of the disorder is not known, etc. The contribution of factor is discussed. That's the main, let's say, philosophical problem with degenerative diseases in general. This is, for instance, a famous observation from America in a very large family with typical Parkinsonism. Some patients were more or less demented, some were not at all. And in 4 cases of this huge family with Parkinsonism, I should say, then one patient had Lewy bodies, here shown here. A Lewy body, for those who don't know, is a histological stigmata, which is supposed to be characteristic of the disorder. This is what I will discuss. But in another patient, the Lewy body were distributed in the cerebral cortex. In another patient from the same family, he had no Lewy body, but it was a tauopathy with neurofibrillary tangles. And finally, in last patient, there was simple neural degeneration without any histological stigmata of the Lewy body type. So this is something strange, isn't it? The same is true for Alzheimer's disease. I will give you my own personal opinion. I am certainly wrong. In some cases, there is no well, you can be certain that a defect, for instance, in the beta amyloid is responsible for the disorder, for instance, because we know this family with a mutation in the A-beta gene. But my idea is that in most patients, the histological stigmata appear as a consequence of nerve cell death, nerve cell dysfunction, I would say. This is shown here. I'm sure you all agree that, for instance, akinesia does result from a very selective of the nigrostriatal pathway. This is because we have the models in rats, in monkeys, and we know some patients with very selective lesions. So there is no doubt that, in general, this is true for all the neurodegenerative diseases, the symptoms result from the cell dysfunction first, very important, dysfunction, loss of terminals of connection before cell death, and cell loss. You certainly agree that the loss of cells in the brain of a patient with Parkinson's disease, but again, it could be the same for Alzheimer. This is caused by several causes. as I said, we will see, in let's say 3, 4, 5% of the cases now, we know that a mutation is mainly responsible for the disorder, so that the genetic factors, for instance, are absolutely crucial. But the real problem is that in the brain of a patient, we found these Lewy bodies, these histopathological stigmata, which, when I was young, was supposed to cause the disease. And this is still true with alpha-synuclein. But the relationship between this loss of cells, of neurons in this case, and histopathology is not clear. For me, at least. I don't know whether it's clear for you, but this could be a matter of discussion. Is the cell loss caused by histopathological corpuscles, such as Lewy body, or is it the reverse? That is, the loss versus the dysfunction of neurons results in the appearance of Lewy bodies, of alpha-synuclein, if I may say so, perhaps. So this is a very complex problem. It's not solved. Personally, I think it's both. There are some cases where it's cell loss, some cases where it's histopathology, and perhaps there is a vicious circle. Anyway, but this is just to show you that we have problem for research in the field of genetics and environmental factors that I will discuss now. It's very rarely hereditary. That's not true. This is not a complete list of the different mutations found in a few families, but in some cases, indeed, you have dominant Parkinson's diseases. You have recessive Parkinson's diseases. And the real problem is what is the role of the innate and the environment. As I said, in few cases, it's genetic defects, almost pure, like in Huntington's. If this is the loss of dopaminergic neurons here, this is time, then with normal aging, do you think that normal aging is characterized by the loss of neurons? Personally, I don't think so. Personally, I don't think so. I think aging is characterized by the loss of terminals. This is also a matter of discussion. But let's say that you have, usually it is the studies made so far show that in 1 century, the neurons which are present at birth and which survive during 1 centenary are lost by 10%. But in Parkinson's disease, of course, when you have a given mutation, a production of the insufficient production of an endogenous metabolite is produced. This is a purely genetic disorder. But in other cases, you may think that it's the disease is due to an exogenous toxic. It can be a chronic exposition to a chronic intoxication. It was said in the past that manganese was provoking Parkinson's disease. I've seen several cases. It's really not similar to Parkinsonism. Nevertheless, this is classical. Or neuroleptic drugs, if you mean, but in case of neuroleptic drugs, cells are supposed not to die. But in other cases, you may have an endogenous toxic which is damaging the cell genome, preferentially at the level of the dopaminergic neurons, for instance. I give you my own feeling, again. I said that I wanted to provoke you a little bit. What you can think in general is that in the 70% of patients who have classical Parkinsonism and who do respond to levodopa at start, then it's a mixture of a predisposition to the disease, not one mutation, perhaps 60 mutations. Perhaps you have 59 mutations. I have a 60 and I will develop Parkinson's disease, plus an exogenous toxic. You know that several studies have shown that pesticides, herbicides, could be toxic in some patients. But perhaps it's a toxic which is in the air that we totally ignore. Okay, so I'm simply posing the question to you. Finally, we should slow down the progression of these as the main mechanism of nerve cell death have been identified. That's absolutely not true. And what is even worse, one is waiting for the success of cell and gene therapy, and I will make an abrupt provocation. So, the mechanism of cell death. We know that these neurons, the dopaminergic neurons at least, are very vulnerable per se, any kind, in the human brain. Why? Because they are free radical bombs. We showed in the past with Pepe in particular, in monkeys, but in humans, that the SOD, the glutathione peroxidase activities in these neurons were defective. There is an enormous accumulation of neuromelanin in these neurons, which can produce free radicals. The presence of iron, huge concentration of iron in these neurons compared to other neurons in the brain. Something which is less known, and we'll come back on that. These neurons are, the most vulnerable dopaminergic neurons are not surrounded by glial cells and vice versa. So that I assume, this is what we showed in the past, that glial cells, through the production of trophic factors, were able to protect these dopaminergic neurons from death. We know that the initiation of neuronal dysfunction, of dopaminergic dysfunction, has been described for years and years in my lab, and totally useless studies, not totally students, but no real discovery. Abnormality in the entrance of calcium, anomaly of the mitochondria that you discussed previously in this audience, overproduction of free radicals, abnormality of the proteasome, the presence of alpha-synuclein, and perhaps I think that the most interesting idea that Parkinson's disease is like a prion-like disorder, inflammation described in particular in this country. And something, but altogether, that's also one myth. We have described since 30 years so many abnormalities, and we are unable to understand what is primary, what is secondary. If this question has a sense, And finally, I would like to indicate to our scientists, colleagues, that we need also always to keep in mind that the dysfunction of dopaminergic neuron is associated with neuronal compensation. At the biochemical level, pre- and postsynaptic receptor overactivity, the production of trophic factors, but also from an anatomical point of view, and it has been shown in many pathological studies that you can see in the brain of these patients some types of synaptogenesis, axonal growth, and an enormous accumulation of glial cells, of astrocytes. And these astrocytes are supposed to play no role at all. In fact, I'm sure that these glial cells are playing a very important role. So you see, we know a lot of description, but no real target for medication. My own idea, that's not my idea alone, but my own ideas are that indeed, if you want to stop the cell dysfunction, or to slow down the cell dysfunction, then you will probably have to use several drugs acting at different levels of this transduction system within the cells. But it's easy to say. It's not easy to prove. Finally, I would like to say, and this is a real provocation for those of you who are interested in cell transplant and gene therapy, which I think is so important to do. Nevertheless, as a provocation, I would like to give you this idea that, in fact, these simple therapy, because it's simple, have no much chance to succeed. Why? Again, it's a provocation, right? But I think it's interesting to keep this in mind. The reason essentially is that the human brain is very complex. To count one neurons of these 100 billions of neurons, if you use one figure by one figure, 1 second per figure, then it should take several hundred millions of years for one person to count. It's enormous. It's enormous. And it's also something which is interesting, by the way. It's that in the substance nigra you have 400,000 neurons. That is 1/10,000th of the whole neurons of the brain. And just to get rid of this pathway, of this highway, I would say, gives you one of the most terrible disorders among degenerative diseases. So it's very complex. And just to give you an idea, in 1 cubic millimeter of the substance nigra, you have 10 to 50,000 neurons. But one dopamine has about 100,000 contacts, 50,000 to 100,000 contacts. So which means that in 1 cubic millimeter of brain tissue, you have about 500 million of connections. And I really have some difficulties thinking that just introducing nerve cells, or genes in 1 cubic millimeter, that is 1,000 cubic millimeters, you will get results. What you will say is that with DBS in the subthalamic nucleus, you are introducing an enormous electrodes, and then you can indeed improve the patients. So I agree, I don't want to make too much provocation, but I think one has to keep this in mind. And finally, we are always discussing with neurons. We totally forget about astrocytes, not those who are in an activated form, but those who are simply astrocytes there. And you have to keep in mind that, for instance, in the nigra, in the ventral tegmental area, you have 6 to 8 astrocytes for 1 neurons. In the lateral part of the substance nigra, remember, Trini, you have only 1 glial cells for 1 neurons. As I said, as if they were less protected, you have to keep in mind that dopaminergic neurons start to die in the lateral part of the... So I will leave you with this. Perhaps, do I have 1 more minute, Jose, or not? Half a minute. So I will stop there, and I will be glad to answer your question. And I would like, again, thank you very much for this invitation, and thank you for your attention. Thank you.
Thanks very much, Yves. Just a few questions, a couple of questions or so, just to take advantage of Professor Agid. Tan ciego. Espera. Vamos a hacer de todo por el presupuesto.
Thank you, Yves. You show us that all of us, we are wrong in our, let's say, traditional thinking. But what do you think about new directions or new ideas to pursue? So which direction should we follow according to your background?
Thank you for this simple question. As I said, clinicians have to describe very carefully the different forms. This is what they did during the last 30 years. I think one of the main problems for me is a relationship between cell death, cell dysfunction. Cell dysfunction is more important because when the cells are dying, it's finished. And the histopathological stigmata. This is not clear for me, and I don't know how it is here, but in my country, during the last years, really, I've had a lot of problem with our colleague in histology because they think that Parkinson's disease is a Lewy body disease, and they, as well as Alzheimer's disease being a disorder of senile plaques and neurofibrillary tangles. And it's not clear. Several experiments should be done in the field. A third direction for me would be the problem of astrocytes. You see, I think that both for function, I give you my idea, sir, but perhaps it's totally wrong. But if I had to start something, I would do that. I think that neurons are routes, avenues, streets. And I think that what is important to survive and to think also are the astrocytes. But perhaps I'm totally wrong, so that's the idea. But you have to, you are asking for ideas. Here are 3 ideas.
Very good. ¿Alguna otro comentario o pregunta?
Thank you. I was very much interested in your ménage à trois last graph, and because traditionally I'm thinking in associations which are ménage à deux, and it's well known that Alzheimer is associated to vascular disease, and that the deposits of proteins are similar in the brain and in the vessels. And Creutzfeldt-Jakob has also a vasculopathy which is linked to PRP. The situation is different in motor neuron disease or in frontal lobe dementia, but it seems that in Parkinson there is no associated disease. And the question is, why do you think that this is like that, if this is like that?
You should know Dr. De Pedro is epidemiologist mainly, so he makes associations.
You see, we, in the past, I think it's a short paper in the Lancet a long time ago, we made a very careful analysis of the density of vessels within the Niagara in controls and in Parkinson patients. And interestingly enough, the number of capillaries were markedly increased in the Niagara. Nobody followed this idea, including people in my lab. But I think that's, if you want an idea, I think it would be nice to, exactly as what has been done quite nicely, but for the other reasons in Alzheimer. I think it's, why? I give you the example of Madrid. You have neurons, no, I should say streets, Calais, Paseo, all over. Does this explain the economic, the cultural, the politic function of the city? No. It's obviously important because if you cut all the street, if you suppress all the street, the Madrid cannot survive. But what is important is what happens in the buildings, in the ministries, in your house, because you are thinking there. So I think that the buildings are the glial cells. But it's a provocation, it's just a paradox.
We thought the glia was like the garbage system, shaking away. So. Very good.
Okay.
Merci beaucoup.